Reminder, if you have not yet given me your mitotic index report, I need that by 2 o'clock today because that's when I'm leaving campus. So after that, it's late. Other reminder, of course, is our final is Tuesday in this room at 10:40 technically we'll start. Goodness, all of your lines. Every last one of you. Anyway, Tuesday at 10:30, so feel free to show up at 9 so that you'll be on time. But we'll start the exam at 10:40 on Tuesday. Need a pencil. Okay, our last topic is meiosis.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Okay, so that's what we're going to kind of walk our way through this morning. Okay, a little bit of history to start us off. Back in the day, hundreds and hundreds of years ago, before we understood all of this, it was commonly believed that it was the male who, through the sperm, put the new offspring into the womb of the female for essentially incubation. And even with the invention of microscopes and when people started looking at sperm cells, they thought if you squinted really hard and you looked at the top part of the sperm cell, you could actually see the little teeny tiny human that was in the head of the sperm. And that little teeny tiny human is called a homunculus, literally a tiny human. So an interesting little historical note. That is, of course, not how it works. Okay, so you should write that down. This is not how it works. But that is what people thought at one point, which I always find a little hilarious, but in some sense, it makes some sense, I guess, historically. Okay. We now know that that is not the case. We now know that sperm and egg contribute equally to the genetic constitution of the next generation. Okay, so here we see lots of sperm competing to get to that first, to be first to fertilize that egg. Remember we talked about the slow-blocked polyspermy a week or so ago. Okay, so there's a method to keep all of those sperm from fertilizing, so we ideally only want one. But there's really our setup for what we need meiosis for. If humans have 46 chromosomes, and we reproduce this way, where a sperm cell fertilizes an egg cell, we need to do something about that number, or otherwise we'd have like a zillion chromosomes by now. Right? And that's really what meiosis is going to do, is going to reduce the total number by half. So that's kind of described here. So, and we'll get to diploid and haploid in a second, but we saw the other day with mitosis, we started with a cell with X number of chromosomes, and we ended with two cells with that same number of chromosomes. That's not going to happen here. And the cells that give rise to sperm and egg are going to go through this specialized division called meiosis, and we're going to reduce the number by half. Okay, so if there were 2 chromosomes to start, then there's only been 1 chromosome in the egg, and if there were 2 in the father, there's only going to be 1 in the sperm. Of course, humans are going to divide 46 into 23, and then a sperm cell with 23 will fertilize an egg cell with 23, and then we're back to 46. So that's how we keep the chromosome number constant within a species. And we're going to use these terms haploid and diploid in just a second. So this meiotic process is going to be all about chromosome pairs, right? Wednesday, when we were looking at mitosis, we didn't care about chromosome pairs. We cared about each chromosome individually, right? You wrote that down and underlined it. Okay? Today, all about the pairs. Okay? So we knew that already. We talked about chromosome pairs, I think, earlier in the course. But these pairs are usually referred to as homologous pairs. And we can visualize that by creating these karyotypes type of pictures. So we each have 2 copies of each of our chromosomes. So humans therefore have 23 pairs. Right? 22 of them are numbered, and these are called autosomes. And these are the same between males and females. So all of us in the room have chromosomes 1 through 22. And then the final pair is the sex chromosomes, the X and the Y. And this is where males and females differ. If you have 2 X chromosomes, then you are chromosomally female. If you have an X and a Y, then you are chromosomally male. So this particular karyotype, with 2 X chromosomes there would be a female karyotype. And these are organized, as you might already know, from largest to smallest. So chromosome number 1 is the largest chromosome, and up until, well, by now, probably close to 10 years ago, we thought chromosome 22 was the smallest, but now that we have all of them sequenced completely through the Human Genome Project, we discovered a little hiccup in this. So it turns out that even though 22 under the microscope looks the smallest, actually chromosome 21 is actually the smallest chromosome, but this numbering system had been in effect for like 100 years at least, and we're not going to go and start changing the numbers of chromosomes, so it's just a little historical hiccup. But in case you're ever on Jeopardy, the smallest human chromosome is actually chromosome 21. And we can do the same thing for males. So here's a male karyotype. And how do I know that? Because there's an X and a Y. So even though the X and the Y are quite different, they do act as a homologous pair in males for the purpose of meiosis. Okay, and these are called homologous pairs because they contain the same genes in the same order. Now they might have different alleles. Remember this from 110, right? Capital A, capital B, small b, those kinds of things. So they could have the same allele. So in this example, 2 capital A's, so they're homozygous dominant. Or they could have 2 copies of a recessive allele, like the B example, 2 small b's, so homozygous recessive. Or they could have one of each, a capital C and a small c. And if you remember, that's called heterozygous. But regardless of the allele, they have the same genes in the same order. That's what makes a homologous pair a homologous pair. So we have 2 copies of each gene on 2 copies of each chromosome. And when we have that situation, which is the situation for basically all of our body cells, that situation is called the diploid situation. We have both copies of each chromosome. So for humans, that would be 46. If you're a yeast, that would be 16. If you're a fruit fly, it's 4. If you're a dog, it's, I'm going to say 72. It's 72, 74, or something like that. So if you have both copies, both homologous, both members of each homologous pair, and you have the diploid situation. Like I said, that is the vast majority of the cells in our body. So skin cells, brain cells, liver cells, you name it, both copies of each pair, so we are diploid. But in order to do this sexual reproduction idea, we don't want sperm and egg to have 46 because, again, we'd have a zillion chromosomes by now. So we need to cut that in half. And when you only have one member of each chromosome, that is called the haploid situation. So only one copy of each chromosome. And as we'll see, that's what meiosis is going to do. It's going to enable sperm and egg to have not a random set of 23, if we go back, so it's probably better, but specifically one member of each pair. So one number 1, one number 2, one number 3, one number 4, etc., through 22, and then one of the 2 sex chromosomes, either an X or a Y. Okay. So that gives us the 23. And then when sperm that has one member of each pair gets together with an egg with the other member of each pair, boom, we're back to being diploid, and we have both members of each pair. So that's kind of the goal, or the major goal, of meiosis and sexual reproduction. The other big thing that we're going to see is that by doing this, from starting with a diploid cell and going to a haploid sperm or egg and doing this whole sexual reproduction idea, well, actually, even before that, this process of meiosis going from diploid to haploid is going to create genetically different cells. There's going to be a number of ways that accomplishes that. But that's a really important idea for sexual reproduction. Presumably the whole point of sexual reproduction is to create genetic diversity, right? So that the next generation is genetically different from the previous one, and then natural selection can work on that and see if the new combination of genes works better than the previous generation it would be selected for. So that is not what we saw on Wednesday with mitosis, right? We made genetically identical copies of cells through meiosis. I always call it the Xerox copying method. Meiosis is not that at all. Okay? We don't want genetically identical cells, we want genetically different cells. There are going to be a couple of ways that meiosis has to do that. And then another big difference between mitosis and meiosis is that in order to do this, to start with one diploid cell and get us to a completely finished sperm or egg is going to require actually 2 divisions, which we did not have for mitosis, right? We just divided the cell once. But in meiosis, the cell divides actually twice. You go from 1 to 2, and then each of these cells divides again into 4. So we start out with 1 diploid cell and end up with 4 genetically different haploid cells through this process of meiosis. So in order to keep kind of everything straight, we call the 2 divisions, or we add on to each division a Roman numeral. So we talk about meiosis 1, which is the first division, and meiosis Roman numeral 2 as the second division. So as you kind of saw in that first animation, it's actually the first division that kind of does what we want. It takes the diploid cell and makes it haploid. So this is usually referred to as the reductional division, because it reduces the number of chromosomes from, we're going to stick with the human example, 46 to 23. So then, of course, we all sit there and we say, well, if that did what we wanted, how come we need the second division? Well, this slide gives us an idea of why we need that second division, because while the first one does divide the number of chromosomes by half, each chromosome at the end of the first division is still made up of 2 chromatids stuck together. And that's not what we want. Okay? We don't want 2 copies stuck together. We want each individual copy separate from each other. And that's what the second division does. And the second division is sometimes referred to as the equatorial division. And this is just going to separate the 2 chromatids from each other. And that's how we go, again, from 1 to 2 and 2 to 4. Okay, so there are going to be similarities and differences. We're just kind of focusing on the differences here. But the similarities are, we're going to talk about the same sort of subphases. So we're going to talk about prophase, prometaphase, metaphase, anaphase, telophase. But again, we have to do it twice, so you just stick Roman numerals after each one, so that you can just distinguish, say, prophase 1 from metaphase 2, for example. Okay, so we just do that simply with the Roman numerals. Okay, so far so good? Okay. So, let's start the process. Okay, so let's actually go back to this one. Okay, so here's our initial diploid cell. So the example here is more like a Drosophila cell with 4 chromosomes, but it doesn't matter. So we have 4 chromosomes, 2 pairs. Right? So we have a red pair. Well, actually that's not true. We have a big pair, the large ones, a red and a blue, and a small pair. So in certain specialized cells, in the ovaries of females and the testes of males, we have this meiotic process going on. So no other cell type does it. Your brain cells never do this. Your skin cells never do this. Your liver cells never do this. It is only specialized cells in the testes and ovaries that go through meiosis. So they go through a regular cell cycle. So they go through G1, S, and G2. So during S, each of those chromosomes is copied. Okay, so we have 2 identical chromatids stuck together. Presumably, if they didn't do that, we wouldn't need the 2 divisions. But for whatever reason, evolution decided, no, let's keep the cell cycle the same, and therefore go through S phase, but it really is that part that makes meiosis a little complicated. But we have to live with what we have. Okay, so it's gone through G1, S, and G2. It's entering the M phase. M phase today is meiosis rather than mitosis. So we're in prophase 1. Now here is where there is a huge amount of difference between mitosis and meiosis. So much so that we subdivide prophase 1 into these 5 stages: lectitine, zygotine, pachytine, diplotine, and diakinesis. And so this is a majorly different phenomenon for meiosis than it was in mitosis. in that a lot of extra stuff happens, and that's what we're going to look at here. However, in general, it's the same thing, right? What happens during prophase? Chromosomes condense inside the nucleus, right? The nucleolus disappears. The centrosomes are moving to opposite ends of the cell. So in that sense, it's exactly the same as mitosis. But a bunch of extra things are happening in meiosis here. And again, it's important enough that somebody at some point said, yeah, we need to name these things and subdivide it this way. The major thing that happens here in prophase 1 of meiosis that does not happen during mitosis is the idea of either recombination or crossing over. Pieces of homologous chromosomes can swap locations with each other. That's crossing over or recombination. And in short, what that's going to do is increase genetic diversity. Now, just believe me now, we'll go through an example after we get through the whole process. But I don't want to bog us down with this right now. But it's an important process to create genetic diversity. I will show that to you more clearly in a few minutes. It is mechanistically a very complicated process. We are not going to look at the actual process step by step. We're just going to kind of walk our way through it in terms of these stages. I think that's more than enough for this class. Okay, so what's happening during prophase 1? Well, the first part, called leptotene, is pretty much just your standard prophase kind of thing. The chromosomes are condensing, the nucleolus disappears, out in the cytoplasm, although they're not showing it to us, the centrosomes are moving apart. So the first part here, leptotene, is your standard prophase kind of stuff. But starting with zygotene, here's where we see the difference. Remember, in mitosis, each chromosome works or acts independently. That's not going to be the case here in meiosis. Meiosis is all about pairs. Starting in zygotene, the homologous chromosomes start pairing up. And they start pairing up by the creation of this protein complex called the synaptonemal complex. Synaptonemal complex. And it kind of starts building at one end and moves up the length. It's kind of like a zipper. Right? So as you pull a zipper up, the two pieces stick together. Right? That's kind of exactly what's happening here with the homologous chromosomes. They're sort of zippered up from one side to the next, and we get them very, 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 very close together. That process is called synapsis. Think it's the same word, same basic word as a synapse between two nerve cells, because they're really, 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 really close together. Same idea here, but we're bringing homologous chromosomes together in this case. Okay, so we're bringing them together, that synapsis, and they're being brought together by this protein system called the synaptonemal complex. Okay, so that's happening during zygotene. The actual crossing over process takes place during the next stage called pachytene. And again, I'll show you a more detailed example of this when we get done the whole process. But you can kind of see here that a piece of the blue chromosome is now attached to the red chromatid, and the part of the red chromatid is attached to the blue chromatid. That's basically what this recombination means. Sections of homologous chromosomes cross over. They detach from where they are and reattach to the other chromatid. And again, just believe me for now, and I'll show you more clearly in a few minutes, that will create a huge amount of genetic diversity. And that's the whole point of meiosis. Okay, so the actual crossing over happens here in pachytene. And then we kind of have, the next two phases are sort of the reverse. So during diplotene, this synaptonemal complex breaks down. Okay, so we don't need it anymore. We needed them really close together so we could do the crossing over. now that that's happened, now we can get rid of the synaptonemal complex. So that's what's happening during diplotin. But you can still see where the chromosomes have crossed over. Okay, so even though the protein complex is breaking down, you can see where they've crossed over. Those places are called chiasma. So as usual, a lot of vocabulary. Okay, so these are sort of the leftovers or the remnants of the crossing over event. Okay, so that takes place during diplotin. And then finally, diakinesis is sort of the rest of prophase. The chromosomes are continuing to condense, and they're ready for prometaphase. Basically, they're ready for the next phase. Okay, so there's a lot more going on here in prophase 1 of meiosis than is happening during prophase during mitosis, like we looked at the other day. Okay, so you want to know these 5 stages: okay, leptotene, zygotene, pachytene, diplotin, and diakinesis, and what's happening during each one. Okay, so this is completely unique to meiosis, and specifically prophase 1. Okay, when we get to prophase 2, none of this happens. Okay, so this is a unique set of events for prophase 1 of meiosis. And again, if it's not clear what all this really means, that's okay. I'll show you that example in a few minutes. But just hold that this is creating genetic diversity. And I'll prove that to you in a bit. Does the general idea make sense? Okay. Okay. But the other thing that it really does is it gets these homologous chromosomes together as their pairs. Right? So this is really where that is set up as well. And they're going to continue to act as pairs throughout at least the rest of meiosis 1. Okay, so just some other visual evidence. So we have visual evidence for this synaptonemal complex. Again, it even kind of looks like a zipper here. And again, this is happening during between every pair of homologous chromosomes. Okay, so we have this crossing over event. So here in this drawing here, there's 1, but then in this actual micrograph, there's actually 3 crossovers, one there, and one there. And these chiasma are clearly visible. Okay, so again, we don't make all of this up, just something. I'm only kidding. That was at least a chuckle's worth of joke there. Anyway, okay, so we have 3 crossovers in this particular setup. So there can be, the other thing to realize is that this crossover process that we've been talking about is random. So exactly where this happens is completely random. And how many there are along a particular chromosome is pretty much random. So in this case, there were 3. Maybe in another cell there's only going to be 2. Maybe there's 4. So all of this process is pretty much random. Okay, but we can visualize these things. Okay, so after prophase 1, what is really holding the chromosome pairs together are these chiasma, okay, until anaphase 1, where they're pulled apart. We'll get to that in a second. Okay, so a much more extensive prophase 1. So unlike prophase of mitosis, prophase 1 of meiosis takes a bit longer because we have a lot more to do here than we do in mitosis. Okay, but after that, the rest of it is somewhat the same, at least again in general theme. So they didn't draw in prometaphase for some reason, but prometaphase 1 happens and the nuclear envelope breaks down. Right, so that's the same. Metaphase 1 happens, and in general it's the same idea. The chromosomes line up at the center of the cell, but with the important difference, here in meiosis, they don't line up individually, they line up as pairs. Okay, so if we were to compare it at this point, in mitosis, it would look like that. The chromosomes would be lined up individually. But here, as you can see on the screen, they're lined up as 2 pairs. That is really the huge hallmark difference between mitosis and meiosis. Chromosomes act individually in mitosis, as pairs in meiosis. How do they get to be pairs? Through all that stuff during prophase 1. Okay, so that's where they pair up. Okay. But this now sets it up for reducing the total number. What's going to happen next? Well, we're going to start pulling things apart. But instead of individual chromatids, we're going to, anaphase 1, we're going to pull apart the pairs. Well, if we're separating the pairs, then that's going to reduce the total number of chromosomes, okay, which is, again, what we want here. Okay, so that's going to happen in anaphase 1. So instead of pulling apart chromatids individually of each individual chromosome, we're going to separate the pairs of chromosomes. So one member of each pair, they draw them up and down, goes up. The other member of each pair goes down. So there's a small chromosome and a big chromosome going up, a small chromosome and a big chromosome going down, or left to right as I drew it here. And then telophase 1 happens. So the nuclear envelope reforms around each set of chromosomes. Nucleoli reappear, etc., and we get cytokinesis, so that one cell becomes 2. But they are not identical anymore, right? We started with one diploid cell with 4 chromosomes in it, in this example up here, and now we finished with 2 cells with only 1, 2 chromosomes. We have reduced the number by half, right? From 4 to 2, or if this were a human cell, from 46 to 23. So again, we'd love to be done now, right? But we can't be done because each chromosome at this point is still composed of 2 copies. That's not what we want. We want each chromosome to be composed of only 1 copy. So that's why we need the second division. So it's all because the original cell went through S phase. I really wish it didn't, because now we'd be done. But it did, so we're not done yet. Okay? But does everybody see how we've now gone from diploid to haploid? So we've reduced the number of chromosomes, total number of chromosomes, by half. Okay? So that's really the critical idea. Okay. So this bottom part here is meiosis 2. We're starting with those 2 haploid cells. Okay? With 1, 2 chromosomes. Well, at this point now, we've already separated the pairs, so there are no more pairs. Right? We got rid of those in the first one. So, if you kind of look at this bottom figure, it really looks pretty much exactly like mitosis. And it is, except for one important difference. The cells that we're starting with are haploid. Otherwise, meiosis 2 is mitosis, because there are no more pairs to deal with. So when we line them up at metaphase 2, they have to line up individually because there's no other way to do it. Right? We already got rid of the pairs. So they have to line up individually. And then anaphase 2 separates the chromatids, just like it did for us on Wednesday. And then telophase 2 and cytokinesis 2, those already haploid cells divide again. So we go from 2 haploid cells to now 4 haploid cells. So if I didn't tell you that this cell was supposed to have 4 chromosomes in it, and I just showed you this bottom part, I bet you'd say, oh, that's mitosis. And you'd be right, in the largest sense, except that these 2 starting cells were made from meiosis 1, so they're haploid. Okay, so this is not, we don't call this mitosis, we call it meiosis 2, but it is essentially a mitotic division in that sense. And again, the other big thing that you can't see in this particular figure is that these 4 haploid cells are genetically different from each other. Again, I'll show you that more clearly in just about 2 more minutes. So we kind of rush through meiosis 2 because it's very much like mitosis. So we started out here, a few slides ago, with 1 diploid cell with 4 chromosomes in this example. by the end of meiosis 1, we have 2 haploid cells, because we've gone from 4 chromosomes to 2, but the 2 chromosomes still have 2 chromatids attached. Those are separated in meiosis 2, so now each of these haploid cells still has 2 chromosomes, but each chromosome is composed of only 1 copy, only 1 chromatid, and that's always kind of what we want by the end of cell divisions. And again, these haploid cells are not genetically identical. More on that in just a moment. Okay. Does that kind of make sense? Okay with meiosis 2, even though we want to do that quickly? Okay. Again, this is just to show you that we don't make this stuff up. We can see all this stuff happening in the under the microscope, so we can visualize all of this chromosome movement and all the pairings and all of that kind of good stuff. So this compares mitosis and meiosis, okay, with, again, a cell with 4 chromosomes total. Excuse me, with 4 chromosomes total, okay, 2 pairs. So again, if the other day when we did that through mitosis, we simply copied them. Each individual chromosome lined up at the center. We separated the 2 chromatids, and we had 2 identical cells, 4 chromosomes, 4 and 4. That's pretty straightforward. But here, we start out with 2 pairs. We line them up as pairs. We separate the pairs, and that's how we go from 4 chromosomes to 2 in each of these cells, a big one and a little one. But each chromosome is composed of 2 chromatids. Those are separated at meiosis 2. So we go from 2 haploid cells to 4 haploid cells, each with 2 chromosomes. But each chromosome only has 1 copy. And again, that's what we want. Okay, so meiosis takes longer because you have to go through 2 cycles or 2 divisions. We have an elongated prophase because of all the crossing over stuff that has to happen. So meiosis, on average, takes a longer time to finish than does your typical mitosis. Okay. So, I've been harping on this idea of genetic diversity. How does that work? Well, before we even do the crossing over, we can get genetic diversity just from the idea of independent assortment. So think back to the genetics portion of Bio 110, right, where we talk about independent assortment. So if you have if we just look at 2 chromosomes on, 2 genes on 2 different chromosomes, and we make this cell heterozygous for each one, so capital Y, capital R, small r, how the chromosomal pairs line up at metaphase 1 is also random. So in the left-hand version, the small y and the capital R chromosome are on the same side, and the capital Y and the small r chromosome are on the same side. So when we go through the rest of this, then we're going to have capital R small y cells, or small r capital Y cells. But in another cell that's going through meiosis, they might have lined up this way, where the small y and the small r are on the same side, and the capital Y and the capital R are on the same side. And then we get small r small y cells and capital R capital Y cells. So that creates genetic diversity depending on how chromosomes line up along the metaphase plate. So even if we didn't have any crossing over, okay, we just had independent assortment of 23 pairs of chromosomes instead of just 2 pairs, okay, that gives us 70 trillion different gametes that each one of us in this room can create. That's not too bad. Okay, so even if we had none of this stuff that we're going to talk about in a second, just independent assortment, each of us can create 70 trillion genetically different sperm or egg. Not too bad. But apparently evolution said, nah, that's good but not good enough. So I'm going to create crossing over, which is going to make the numbers ridiculous. So, let's look at this crossing over idea more closely. Okay, so back in prophase 1, we had homologous chromosomes getting together, right, through synapsis by the synaptonemal complex. I'm going to leave a little bit of space so I can put letters in here. Okay, so here's one pair. There's one homologous pair. Let's put a bunch of genes on this. And just for simplicity, we'll make one chromosome have all the dominant alleles, so all the capital letters, just to make this simple or simpler. And we'll make the homologous chromosome have all the recessive versions. Okay, so there are two identical copies because the cell or the chromosomes have gone through the S phase, so each one is composed of two identical copies. Okay, we're in prophase 1. The synaptonemal complex has joined these two together. The crossing over process is happening. All of the different enzymes that are necessary for that are coming along. And let's just say we have a crossover here between E and F. Why? Just because I picked it. We could have done it between A and B or H and I. Remember, that's random. So let's say this in this particular cell, it happens just once between genes E and F. Now, it'll be simpler if we number the chromatids so we can kind of keep track of them. Now, the other thing to realize is that crossing over can happen between any two non-sister chromatids. So 1 and 2 are sister chromatids, 3 and 4 are sister chromatids. Right, we talked about that the other day. Any non-sister set of chromatids can go through this. But I'm not a very good artist, so I can only really draw it effectively between 2 and 3. But it can happen between 1 and 3, or 1 and 4, or 2 and 3, or 2 and 4, any non-sister chromatid. But I can't draw those very well. So we'll just do it between 2 and 3. Okay, so if it happens between chromatids 2 and 3, between genes E and F, what do we get? Okay. Well, did we do anything to the first chromatid?
No.
No. So all of those 4, 5, 6, 7, 8, 9 genes stay together. Right? So we didn't do anything to that one. So one cell is going to get this chromatid eventually, with still all of the capital letters still together. Right? So that's the parental version. Right? We didn't change anything around. We didn't cross over anything. So this would be a parental configuration. But chromatids 2 and 3 have this recombination. So they're going to have some of chromatid 2 and some of chromatid 3 now stuck together. So we're going to get capital A, capital B, capital C, capital D, capital E, capital F. I went too far. Capital A, capital B, capital C, capital D, capital E. But then we cross over to the third chromatid and get small f, small g, small h, and small I. And this is a physical process. These chromatids get cut and pasted by the enzymes that do this. So these are going to be physically attached to each other. Okay, so that's not parental. We've recombined, we've crossed over, we've created a new combination of those 9 genes. Same thing for chromatid 3. It's going to be kind of the opposite. Small a, small b, small c, small d, small e. We cross back over to chromatid 2 and pick up big F, big G, big H, big I. And then we didn't do anything to the fourth chromatid, right? So all of those stay together with all the smalls. But now, since each of these chromatids goes into a different cell by the end of meiosis 2, and just looking at this one chromosome pair, each of these cells is genetically different from each other. All right? This has all capitals. This has 1, 2, 3, 4, 5 capitals and 4 smalls. This has 5 smalls and 4 capitals. That has all smalls. So they are genetically different from each other. So if we throw this into the mix, if only one chromosome in humans, if only one chromosome did this, then we go from 70 trillion different sperm or egg to 5 quadrillion, 5 times 10 to the 27th power, whatever that is. But it's not just one chromosome. All of them do this. So at least all of the autosomes do this. So there are on average 22 crossovers, not just one. There's on average 1 per pair. So then it gets even more ridiculous if each chromosome does this once, and that's the average, then each of us in the room could make 5 times 10 to the 61st power genetically different sperm or egg. That is an unimaginable amount of genetic diversity. Okay? But that's what crossing over does. It goes from 70 trillion, which I think is already a pretty good number, to 5 times 10 to the 61st power, which I can't even imagine that number. Okay? So that's really why this happens, is to just make this extraordinary amount of genetic diversity possible within sexual reproduction. Does that kind of make sense? Okay, so this is crossing over this way. I like it with a bunch more letters than this. We'll go from there. Okay, very, very quickly. So that's the general process when it works well. When it doesn't work well, that process is called non-disjunction, where chromosome pairs don't work the way they're supposed to. So we're going to focus on just one pair, okay, that's not going to work correctly. Okay, so what's supposed to happen? We're supposed to, at meiosis 1, separate the pairs, right? So the blue one would go there, the black one would go there. So that's what's supposed to happen. But sometimes they don't separate correctly. So both copies go to one cell, and that cell gets no copies of that chromosome. Now, we're just looking at the one pair that's doing it wrong. Presumably all the others are doing it correctly. Okay. Well, then if this one has no, chromosome 4, then obviously when it divides again, these cells won't have any chromosomes 4, right? Because both of them were in here. Now, when these separate, you're supposed to only have 1 copy, but these are going to have now 2 copies of, say, chromosome 4 or whatever chromosome you want. That's a problem. All of these eggs, in this example, or sperm if it were sperm, are abnormal. These don't have any copy of that chromosome. These have 2 copies of that chromosome. So the earlier the non-disjunction happens, the worse it is. So when it happens at meiosis 1, all of your gametes are abnormal. But it doesn't have to happen early. It can happen later. So this can go through meiosis 1 correctly, and this cell can go through meiosis 2 correctly. So that's perfect. But let's say this cell, for whatever reason, didn't go through meiosis 2 correctly, so the chromatids don't separate. So again, 2 copies go into there, 0 copies go into there. So in this case, in meiosis, if it happens in meiosis 2, then usually only half the gametes are sort of bad in that sense. So this can create, though, problems with chromosome number. So think about, the best example is, say, Down syndrome, right? Where you get 3 copies of chromosome 21. Well, how does that happen? Well, it could be that the egg had 2 copies, and then the sperm brings 1, and there's your 3, or vice versa. It's a normal egg, but you have a sperm with 2 copies of chromosome 21, and then you have 3, and that's Down syndrome. So it creates problems with chromosome number, non-disjunction. And this happens at varying rates. There is a correlation with age, especially more so in women than in men, although there is a correlation with men as well, which is not quite as dramatic as it is with women. So this is the rate of Down syndrome versus maternal age. So you can see after about 35, the risk goes up substantially for a Down syndrome, or to produce a Down syndrome child, because as we age, the process of non-disjunction happens more frequently for reasons that are not perfectly clear. But there is an age-related situation here. And then in the last minute, I just want to introduce the idea of what's happening kind of after meiosis. Okay, so we kind of looked at meiosis in general. This is looking at sperm production specifically. And the one interesting little twist here is that as sperm develop, the cells don't completely split from each other. They have these cytoplasmic, what are called cytoplasmic bridges. So cytokinesis is not complete. And that's probably because, remember males are XY, so some of these sperms have an X chromosome, some of them will have a Y. And it turns out some of the genes that are necessary for sperm to develop are on the X chromosome. So if these cells actually separated completely from each other, the poor little Y sperm, with having no X chromosome, wouldn't have the genes to actually develop into sperm. So they don't separate completely so that the proteins that the X chromosome are making can be shared across all the cells, whether you're X or Y. So whether you're X or Y, you actually get to develop, which is kind of interesting. And then the twist for females, and I'm sorry, I'm going to keep you about a minute over, is unequal cytokinesis. So remember on Wednesday we talked about most of the time the cell splits right down the middle. For eggs, it does not. And that's probably because making an egg is really expensive from an energy standpoint. Making sperm is cheap, which is why males make so many. Making an egg, very, very expensive. Lots and lots and lots of proteins are in the egg, and those are, as we've already seen, are very expensive to make. So, through meiosis 1 and 2, and this unequal cytokinesis, only one of those 4 cells becomes the egg. So most of the cytoplasm stays with one cell after each of these divisions. The little teeny tiny cell that's made is called a polar body. So you only actually get one egg and 3 polar bodies. The polar bodies don't develop, so you only actually make one. Females only actually make one egg after meiosis. Again, because making 4 eggs at a time apparently would be too energetically costly to do that effectively. So there are little twists for sperm versus egg that I just wanted to point out. And there's the course. Okay, so our